Swayam Prabha. Digital India. Educated India.
Okay, so yeah, so as I said, what I'm going to do in the next 40 minutes or so is to give some examples from different, let's say, fields, so as to say, and one such is connection to biology, right? So what I have shown here is, for example, this is a cell, right? It's a simple picture of a cell, right? And if you look at the outer boundary of the cell is what is called cell membrane, right? And this cell membrane, if I zoom in, right? If you zoom in, if I use, some, say, sophisticated techniques, right? If you look at carefully, it basically has what is called as a bilayer, okay? Bilayer because there's one layer here and another layer adjacent to it, right? And these bilayers are basically made up of a molecule called a lipid molecule, okay? It's a fatty acid, right? And this is basically like a surfactant, okay? And only thing is, like surfactant, they have a hydrophilic part and a hydrophobic part, right? So they carry a lot of resemblance to synthetic surfactants, right? And when people have been able to make such molecules in the lab, of course, although they occur naturally in the living cells, people can make such molecules and then if you take these molecules and put in water, they self-assemble and form nice structures like this, which resembles cells, okay, with a bilayer, okay, or a surfactant membrane surrounding a liquid-like core, okay. And so therefore, what people do in the lab is to kind of, chemists think about how to synthesize molecules which are similar to this, for example, and people who work in colloids and interface science, or or people who work in physics and chemistry and other fields, what they do is they study to understand what happens if I put such molecules in water or any other fluid. Will they assemble and form structures like this? How do I create structures like this? How do and what is the properties of these membranes? Is it a stiff membrane? Is it a stuff like that? Okay, people are so therefore one of the motivation for studying colloids basically comes from connection to biology, because people can make molecules which can kind of form cell membranes, and this is an example of a protein or a lipid molecules in the body. As you can imagine, it basically has nitrogen, phosphorus, oxygen, carbon, hydrogen, right? And that's so these linear chains that you're talking about, they're what are called as tail groups, right? There's one tail here and one tail here, right? And because of their inherent property that they have a hydrophobic part and a hydrophilic part, the moment you put them in water, they want to minimize the exposure of hydrophobic region to water. Therefore, they form an assembly where the head group, hydrophilic, is in contact with water, and again the head group on the other side is also hydrophilic and that's in contact with water, okay. They form these nice structures, okay, just by nature, right? Okay, because of the different forces involved, okay, in their assembly, they form these nice structures and they form these bilayers. And people from colloids literature, they try and ask question about how do I make these bilayers, how do I control the size of these structure. For example, people try to measure the stiffness of the membrane by using techniques like atomic force microscopy, some of these sophisticated techniques which gives you information about are all the molecules arranged in a particular way? Are there gap between the molecules? You can ask a lot of questions and and get some insights into some biological processes that are occurring in these systems. And these structures that I said, which are created by putting lipid molecules in water, they're what are called as liposomes. Okay, the reason why they're called liposomes is because they are made up of lipid molecules, and then they organize, form these spherical structures, okay, which resemble, as I said, cells, right? And now, as you can see, there are hydrophilic regions, right? The inner one and the outer one, they are hydrophilic, and the interior is hydrophobic, right? Because it's a tail molecules, right, which only has carbon and hydrogen. They're hydrophobic. So now, one of the nice things about these structures is that people use them as what are called as cargo carriers. Okay, they're used as cargo carriers, and they have a lot of applications in cosmetics, medicine, and genetics, because when I'm making these structures, I can incorporate either a hydrophobic drug or a hydrophilic drug or any complex molecules. So what I'm trying to say is that if you synthesize, if you make these liposomes in the presence of some essential molecules, okay, these structures can be used as a means for incorporating these molecules in these structures. Okay, so for example, this hydrophilic stuff will be inside, because the inside is water, right? And as I said, the inside is all hydrophilic. So therefore, hydrophilic molecules can stay inside, and hydrophobic, right? And the hydrophobic stuff can be in between, right? Because as I said, the inner region is all hydrophobic. Therefore, hydrophobic molecules can stay in the bilayer, right? And hydrophilic can be in the interior region. And something that is a complex structure can actually have small region, in contact with the inner fluid, small region in contact with the outer stuff, as well as, it is something where this both hydrophilic, hydrophobic, right? So some portion will be in contact with the hydrophobic region, and some portion will be in the hydrophilic region, right? So therefore, what people do is they can synthesize these structures in the presence of, for example, let us say that I want to create a cough syrup, right? Cough syrup, and say that the cough syrup is made up of some molecules that you want to take, right? Okay, one way of doing this would be putting it in water, right? I take water, right? Put these molecules and make a solution, and you can have it as a syrup, right? But a simple formulations like that don't work, right? Because you should look at the compatibility of those molecules with the liquid. Okay, now will the liquid molecules properties itself change when I put in water, right? These are all important questions that people ask when they try and design these drug delivery systems, right? So therefore, what people do is they make up these molecules, called these lipid molecules, and I can incorporate whatever active ingredients. Okay, in pharmacy literature, people talk about what are called APIs, active pharmaceutical ingredient. That's a molecule that in whatever tablet that you take, right? That's the thing that is doing the magic to heal you guys, right? So therefore, so when people design such liposomes, I can incorporate these molecules, and I can, if I have for example, a kind of formulation which has these structures, right? And I can use them for transporting hydrophilic or hydrophobic or any complex molecules into either in, for example, when you take it as a syrup, for example, or you could be used in some, for example, cosmetics, right? Very complex formulations which has depending upon what they're used for. Sunscreen lotions, for example, have some high molecules, coexisting with some water and oil kind of stuff, right? So the basically the delivery of essentials, can be done by using these as vehicles for their transport, okay, either into the body or on the, go ahead. So what is the method of incorporating these molecules? Okay, there are few different ways. So one of the simplest way that people are trying to think about is that if I take, for example, the way these structures are made, you take water, okay, and you take a known amount of these lipid molecules, okay, put it in water, and there are cases where such structures can form spontaneously. There are cases where you have to add energy to make these structures. When I say energy, basically mixing it, okay? And typically it turns out that many of these lipid molecules, right, they're more hydrophobic, okay, overall, right? And therefore, when you put these molecules in water, they're kind of insoluble, they kind of float on the surface, right? Right? So when you mix them, what happens is they kind of assemble in a bilayer formation, but these bilayers cannot be infinitely long, right? They kind of fold up and form these structures, okay? So there are two ways of making these structures. One is either spontaneously use the right combination of fluid and a lipid molecule, or you take a fluid, put these insoluble molecules and then mix it up, okay? And when you want to formulate with these structures, what you do is, one of the simpler way of doing this would be that before you make these structures, you incorporate all the essential stuff and then you mix it up, okay? That's one way of doing it. Yeah. Correct. So what is the, okay, that's a good point. Okay, what he's trying to ask is, okay, I've made these things, okay, I'm telling you that, you know, it can be used as delivery systems. How is it delivered, right? There are different ways of, you know, doing these things. One of the simplest way of doing, for example, we know that, you know, there are pH changes in our stomach, right? Okay, what people do is people design molecules which respond to pH, for example, okay? When you do formulate them, you formulate them under certain conditions, so everything is all nice, you know, they are nice spherical structures like this with all these things, you know, in different regions. Then when it reaches a particular location, there could be change in the temperature, for example, there could be change in the pH, right? And then, you know, you can do a pH kind of triggered bursting of these things, for example, okay? And when they do that, they kind of do this. Yeah.
So since the liposomes are spherical in structure, how do two or more liposomes attach to each other?
Okay. So okay, that's a good point. So so her question is, the way I've drawn the schematic, right? It looks like liposomes are spherical structures, right? And her question is, if there are many, you know, such liposomes in solution, how do they attach, right? I must say that, you know, the liposomes need not be spherical. There are, so if you look at, so this particular field, right, of taking surfactant molecules and forming such structures, it comes under a broad field called surfactant self-assembly, okay? You would have heard of something called micellization, micelle formation, some of you may know. So micelles are kind of spherical structures that form when you put surfactant molecules in water and above a critical micellar concentration, okay? Maybe you can read up a little bit about it, okay? So similarly, you can form spherical micelles, you can form liposomes like this, you can make liposomes that are elongated, right? You can form them in many different sizes and shapes, and you know, see, for the two liposomes to fuse, right, you know, come together, there should be, so one of the simplest way to think about this would be that, you know, these membrane that you have, no, it acts like a barrier. If there are two lipid molecules that they come close to, right, there's a barrier because of the membrane, and that basically stops them from fusing together. So typically when you form these formulations, of course, if you change the pH conditions and, you know, temperature and stuff like that, they can fuse and form larger structures. But, you know, you can, at this point, you can think about, you know, there's a barrier and that kind of prevents them from coming together. Maybe at this point, maybe that's good enough for this point. Yeah.
Sir, is it possible to like deliver only the hydrophobic part in a certain area of the body, or like wherever you are delivering, and then like in some other part based on like some change, you would deliver the hydrophilic part?
That's a good point. So the question is that what is it possible to selectively deliver different drug molecules at different locations of the body, right? It is possible. I think, see, people talk about what is called a targeted drug delivery, right? Okay, so that's a huge field where people are trying to come up with some new ways of designing molecules. This may not be the best way of doing it. Okay, liposomes may not be the best way of doing it, because when you want to disrupt them, the entire structure collapses, right? Okay, but you could think about some other cases where I can have a hydrophobic particle with hydrophobic drug, for example, which doesn't care whether the pH is 10 or 12, for example, right? But the other ingredient there, kind of releases. You can do things like that. Yeah. So, there are people who work in the field of materials, okay, where they use a route called colloidal processing route, okay, for making materials, okay. Let us say that I'm just thinking about the best example to make. Best example would be let's say ceramic cup, right? The cup that people use for coffee cups, right? Not the disposable ones, but the ceramic cups, right? So one way of doing that would be what you do is you start with a ceramic particle, and you pour somewhat of water, prepare a sol. Okay, sol is basically particles in a fluid, right? And what you do is now if you look at here, they're all individual particles, right? Now if you look at the next schematic here, the particles are kind of connected, right? So that means you're going from a sol state where the particles are all individual, and you kind of induce what is called the aggregation of particles. That is, the particles come together and they form clusters. That's called as a wet gel. You can think about going from like say syrup to an ointment, right? Okay, syrup is more liquid like, okay, ointment is more like a gel, right? You can think about something like that. And then what you do is you dry it up. Okay, that means I'm removing all the liquid. Then once the drying is done, people do what is called compaction. Okay, and then they do a simple process called sintering. Okay, sintering is a phenomena of heating this material to a temperature close to the melting point of these particles. Okay, when you do that, what happens is these particles fuse, right? And I can shape it into any shape that I want, right? I can make it into a cup, okay, or I can make it into a kind of a slab, for example, right? I can shape it into any structure that I want, then I heat it up, and then I can make a nice final specimen, right? There are people who work in materials engineering where you start with colloidal scale structures, make a dispersion of these particles, aggregate them to make a gel, and then remove the liquid, compact them, heat them close to their melting point, and then I can make structures, right? I can either make a solid structure or I can make a porous structure as well. Okay, depends on how you do compaction and stuff like that. In fact, there's a huge industry where people are trying to look at filtration at high temperatures and stuff like that. Okay, because ceramics are, they're suitable for high temperature applications. I can make a porous ceramic which have applications in, filtration of molten metals, for example, filtration under harsh conditions, right? People kind of use some of these colloidal processing routes in which you start with dispersion of nanoscale or colloidal scale particles and then end up with a specimen of a particular porosities, particular dimension, particular shape, particular chemistry. I can make porous silica. I can make titania-based ceramic structures. I can make alumina-based ceramic structures and stuff like that. Okay, there's a huge interest in studying colloids from the perspective of materials development itself, right? So I work in a field called particle stabilized emulsions. Okay, emulsions, as all of you know, it basically is, so I take water right? And I take oil. If I mix it up, what will happen? If I leave for sufficient time, it will again become water and oil, right? Now what you do is, classically what is done is, before I do this mixing process, you put in some surfactant molecules, right? And what will happen is, if I do that, you can get maybe water with droplets of oil, or I can get oil with droplets of water. Okay, you can create what are called as water in oil emulsions or oil in water emulsions. But if I want to do that, if I do not have surfactant, it doesn't work out. You have to put in some surface active agents, and what these do is they go and sit on the interface, right? They sit at the interface and they can stabilize, right? Now similarly, it turns out that you can also use particles for stabilization. You know, instead of surfactant molecules, if I were to have particles along with these fluids, I do exactly same process. You can get droplets like this, and now what is there on the droplets are, this is a 9 micrometer particle and this is a 2 micrometer particles, right? So that means I am able to coat every droplet, okay? I am able to coat every droplet that is generated during the mixing process by the particles that are there in the fluid, and it forms a nice shell, right? Very simple process, right? I take water, oil and a stirrer and put in a few particles, mix it up. That's it, nothing more than that, right? One of the interesting things about these materials is that because I have these particles on the surface, again they stop the coalescence process, right? If I have two droplets, if they merge, right, the reason why you get a complete phase separation here without any surfactant or particle is because two droplets come, they join, become a bigger drop. Bigger droplets further join, they become even bigger drop until you have a complete phase separation like this, right? Now in this case, there are particles and every droplet is coated with particles, right? And that coating basically stops them from joining or merging together, what is called the coalescence process, right? And you can get nice structures like this, right? Now, what is the application of these things? I talked about liposomes, right? Like liposomes, people talk about what are called as colloidosomes. Okay, these are actually again spherical structures in which the shell is not the lipid molecules but the colloidal scale particles, right? Now what I can do is I can take such droplets, I can compact them and do exactly the same process that I discussed here. I can compact, instead of compacting particles here, right, I can compact droplets with these particles and I can make a porous material like this, okay? So these are very nice routes of making new and novel porous materials which have applications again in many different areas, including filtration of water, for example, right? You can look at many high temperature purification operations that people use are kind of made by such structures. You can make hollow capsules like this, you can make porous materials just by starting with these things, okay? Therefore, these nanoscale particles and nanoscale materials have this amazing ability of stabilizing interfaces, okay, exactly the way the surfactants do, but they offer additional advantages, okay, in terms of better stability, you know, they are more thermally stable, okay, and better control on the loading of, you know, different entities and stuff like that, okay? This is a huge field and this is one of my research areas, and, you know, if you're interested in know more, I can talk a little bit about that. Okay, yeah, some inspiration from nature, right? As an example, what I do is I'm going to show you some pictures of peacock, right? So any thoughts, connection between peacock and colloids and nanoparticles? What is that? Huh? Feathers resemble structure of colloidals. Really? Okay. What are the colors?
Do the colors originate from nanoparticles?
Yeah, somebody says do colors originate from nanostructures? Yes, that's one answer. So it turns out that again there are a lot of nice studies. Okay, yeah. What's that?
There are parts of dispersed color things the whole thing.
Parts of dispersed.
Like collides,
Yeah.
the particles are dispersed in a continuous medium. The same way the blobs of colors are dispersed in a
So he's saying the similar way the particles are dispersed in a fluid, he's saying the colors are dispersed in on the end. Okay, that's the analogy that he's trying to draw. Okay, what is it?
Feathers are hydrophobic.
He says the feathers are hydrophobic. Okay. So what I wanted to mention was about, see, some of the things that you mentioned are partially true. Okay, for example, is more hydrophobic, right, and stuff like that. I just wanted to draw attention to what are called as structural colors. Okay. So the general understanding is when people make paint, right? When people make paint, they put pigment, right? And these pigments are the coloring agents that you put in those formulations. Like I mentioned example of titanium dioxide, right, for white color, and stuff like that, right? But there are colors which purely come from structure, okay? Purely come from structure. What do I mean by that? So if I take peacock, right, and people have done this, okay? There's a very nice article, right, that is PNAS, right, PNAS, right, Proceedings of National Academy of Sciences, right, published, in 2013, right, where what they did is that they looked at, the different parts of the feathers, and the length scale here is about 1 micrometer, right, and the length scale here is about 500 nanometers, and here it's about 500 nanometers, right, and you can see that, all the structures that you see here, they're all colloidal length scale, right? It is between that 1 nanometer to 1 micrometer size range. And in structural colors, the color that we see, it is basically because of the interaction of light with the colloidal or nanoscale structure, okay, on these creatures, and that's what gives us different colors. Okay, for example, there are regions where the arrangement of nanoscale structure is different, their shape itself is different. There are cases where there are pores between the structure. For example, these are all spherical kind of shaped, right? So therefore, depending upon the arrangement of structures and their interaction with the light is what produces these brilliant colors. Okay, and what people have done is, okay, this is another example from for example, the butterfly, right? This is a picture taken from one of my students. So where what he did is, he picked up one of the feathers of the peacock, right? And if you do SEM, right, if you look at zoom in, right, this length scale is about, from here to here is about 20 microns. But if you look at individual structures here, they're again very, very small, right? Therefore, so these structural colors are an important class of materials, okay, that people are trying to not only understand things from the nature, but people are also trying to mimic them in the lab. For example, okay, I think I have an example. So this is another example where this is a bird that is found in some parts of the world, where there's a difference in the coloration here, right? One is both of them are blue but slightly different, and one of them basically has a nanoscale structure like that on the feather, and this one has a more close packed kind of arrangement of spherical structures. That's what kind of gives these structures. Okay, and what people have done is they've done experiments in the lab. Yeah, go ahead. What's that?
Like, is it based on absorption or something?
So all the phenomena such as the absorption, reflection, okay, all that is important. Okay, and what people have done is that so people who work in photonics field would know more about it. But from my what I know is that what people have done is they're able to create specific arrangement of particles on the for example, surface, for example, and depending upon the angle of incidence and and different properties of the light, and the wavelength of the light that people use and stuff like that, you can actually produce different colors, is what I know. Yeah. So this is a simple experiment that people have done. It's a work from Harvard University where what they have done is it's a simple example of drop casting. Okay, drop casting is a technique where I take a drop, put on a substrate. That's it, right? That's called drop casting. What you see is there are 5 different drops. Okay, all of them basically have particles which are polystyrene, and the size of these particles is 226 nanometers and 265. Okay, everything has exactly same concentration of particle and of this size. Okay, but what they do is they add a small amount of carbon black. Okay, carbon black is again a very naturally occurring material, right? You burn stuff, you get carbon black, right? And what they did is they basically changed the concentration of carbon black from 0.02 to about 11.2%. Simple drying process, that's it. Okay, there was some solvent in all these droplets, and the solvent is evaporated because of the the drying process, and then whatever particles, whether you had PS or carbon black, ultimately they kind of deposit on the surface on which it is dried. And as you can clearly see, the color that is generated because of the drying process is very, very different, right? And what they argue is that if I look at, for example, this one, okay, this is a carbon black that they put, and there's a kind of an amorphous, or kind of a irregular arrangement of particles on these deposits, and that's what leads to these colors. Okay, so what people are trying to do is to kind of manipulate the deposition of particles on the substrate, and then try and draw analogy between nature and synthetic colors, and then or not the synthetic colors, but mimicking these natural colors, and how can we create such colors by manipulating particle chemistry, particle interactions, the way I arrange these particles on the substrate and stuff like that. Okay. So the next example that I wanted to quickly touch upon was this very famous example of lotus leaf effect, right? I know how many of you know. Lotus leaf is an example of what are called as self-cleaning surfaces, right? Okay, if you Google up, right, you get a lot of hits on lotus leaf being an example of a self-cleaning surface, because the moment the water drops falls on these leaves, they just roll down. Okay, as it rolls down, it collects all the dust, whatever that is there on the surface of these things, right? And the reason why that occurs is because lotus leaf basically has some micro and nanoscale structures. So this is an electron microscopy image of a lotus leaf. You have these big bumps, right? And also very small, structures here. These are, say, this scale bar is about 10 micrometer. Therefore, there are micron scales bumps, right? Plus, there are nanoscales features, and these features are basically responsible for the lotus leaf effect. Okay, so what it basically means is that when the water drops, right, and water drop doesn't like to be in contact with these surfaces. Okay, I talked about this hydrophobic nature, right? It takes a shape of a spherical ball and it just rolls down. Okay, it doesn't stick to these surfaces, right? And so people kind of draw inspiration from such structures, and you'll see a lot of articles on papers written about making surfaces that mimic lotus leaf effect in the lab. For example, one of the simple techniques that people do is they take a lotus leaf, right, and what they do is they put in a liquid, okay, on these lotus leaf, and they've come up with some ways of transferring whatever structure that you have on the lotus leaf onto the liquid that I've put in, right? I can basically make a mirror image of what you have on the lotus leaf, right, on the liquid, right? And they can use that as a template for again making a lotus leaf surface, right? So people kind of look at some aspects like that. Yeah, so coming back to this example of soot, right? So I said that people have been able to make this soot formation, right? And so if you look at what they did is they make this coating, and they, of course, they do a little bit of chemistry, okay, the details are not important, but what is important is that this is the surface on which there is a coating. You see the contact angle of water is 165, okay, close to 180. Other interesting thing is that they do some manipulation and they're able to create a similar surface where the contact angle is 156, but not for water, but for an organic compound called hexadecene. Okay, that means that the soot formation they exploited to make both surfaces which do not like water and the surfaces which do not like oil as well. Okay, these are, you can think about something like omniphobic, right? They neither like water nor like oil, right? Again, they have again a lot of applications. As I said, you can look at anti-icing surfaces, for example. You can use them, for collecting, oil and water, depending upon, in the surfaces that, that you kind of immerse these surfaces in and stuff like that. Okay, so therefore, there are a lot of people who are trying to exploit the simple, processes, which generate particles, and how can we exploit them in creating this hydrophilic and hydrophobic surfaces. That's again a huge area where a lot of people work on. Yeah, please.
The previous slide.
Sure.
In B it's just a sphere and the surface, but in A there's this kind of blob, so there is anything of significance.
So you mean these two?
Yeah, in A there's this kind of like protrusion.
Oh no, so basically, so you're talking about this, right? Yeah, this is maybe the needle that they used for making these droplets. I think there's nothing more to it. Yeah. Yeah. Okay, so this is a. So all of you know about what are called as phase transitions, right? So all of us know that you take a solid, right? For example, ice, right? And I basically heat it up, it becomes a liquid. If I go further, it becomes a gas, right? And we also know that they have different densities, right? Slightly different, right? At least between these two states, right? Now, and we know that they are more closely packed in the case of solids, right? And here the spacing between the molecules is slightly larger compared to the solid. And then in this case, the distance is even larger, right? People use colloids as what are called as model atoms, right? What do I mean by that? Would be clear if I am going to. So what people do is they kind of use colloids, okay, to kind of observe these phase transitions, right? We know that we learn about these things in textbooks, right? But if I want to visualize it, how do I do? Right? Because these are all made up of atoms and molecules, right? I can't use microscopes to observe these things, right? I cannot use a microscope and then look at the arrangement, where are they located, what is the spatial distribution. I can't do all of that, right? Because they're too small to be seen by a microscope, right? Maybe I'll come back to this a little later. So this is an experiment that people have done, okay, in which what was done is they took a fluid and then they put in particles, and the number that you see here, right, this is 0.494, 0.58, 0.64, 0.74, these are what are called as volume fraction of particles, okay. This is the volume of particles divided by the total volume, okay. You can think about it as a solid fraction, right. If it is 0.5, it has 50% particles and 50% water, okay. If it is 0.74, 0.74 is particles and 0.26 is water, right, or a fluid, right. And what you can see here is that when the concentration of particle is less than 0.494, okay, this is a microscopy image, and you see that the particles are kind of randomly distributed, right, there is no specific order as such. I increase the concentration a little bit further. This is about between 0.494 and 0.545. you see there are regions where they are still randomly distributed, but if you look at a region here, for example, I have one particle 6 neighbors, next particle 6 neighbors, right? There's a kind of a crystalline kind of arrangement. There's a more regular arrangement here, at least in this corner. I go up further in concentration, you see everything has become nicely ordered, right? And if I go further, again there's a disorder, there's a blast-like formation, okay? So what I'm trying to tell you is that I can do a simple experiment in the lab, taking a liquid, putting in particles, put it under a microscope. I can start observing what is happening to the particles, right? I can look at their spatial location, I can look at their arrangement. This is one simple model system where people started with particles, right? They started with a fluid, they put in particles at different concentration, and they saw that you can have particles arranged in a liquid-like fashion, okay, no particular order, or a crystalline kind of arrangement, more close to a solid, and you can do all of this in real time, okay, because with the advance of microscopy, right, people are able to go and look at length scale, which are few nanometers and few micrometers, can be seen very easily in a lab microscope. There are several of them in the institute, including our lab, okay. Therefore, you can watch them live, okay, and then observe all the phase transition that you learn in your 10+2 or lower grades. You can observe them in situ live, and you can look at the formation of these things, right? And people can ask a lot of questions, okay. I start with a structure like this, and how do I go back from here to here? People look at melting, right? We know that melting is basically starting with a solid stuff and then going to a liquid state, right? I can do experiments to look at melting, phenomena, for example, right? I can look at crystallization process. I can go from a liquid to a solid, right? I can do all of that in the lab, okay? And therefore, there are class of people who look at particles as model atoms and then use them as atoms to look at understanding the fundamentals of a lot of physics that happens, right? In terms of formation of solids, formation of crystals, melting processes, freezing, all that stuff can be studied, okay? Therefore, there's a lot of interest in studying such, particulate matter. I've talked about that. There are a lot of nice books, okay, available for you guys to read up about these things, okay. This is basically a nice report by Robert Brown, available freely on the web page. I talked about this Brownian motion, right? This is a nice book by this John Perrin, who got a Nobel Prize that I talked about, right? And there's another nice book called Middle World, okay? Middle World is something, okay, the middle world, the title is because it is neither atom, not too small, okay, not too big, right? Okay, it's a middle world, so that's what the title comes from. And this is a restless heart of matter and life, okay? These particles in the nano size scale, they're never stationary, right? They're always moving, okay? They're always exhibiting Brownian motion. Okay, you can take liquid, put in these particles, okay, macroscopically it looks like there's nothing changing, but there are particles which are always moving, right? They're always restless, right? So therefore, this is a nice book which talks about the history of, the development in the field and, who did what and stuff like that, okay? This is again some nice, okay, I had a video and unfortunately it doesn't play. Oh yeah, it plays, right? So this is a movie of, again, this, I talked about this, the movie that I showed earlier was an animation, but these are actually real particles. These are all the white dot that you see. These are nanoscale particles immersed in a fluid, and as you can see, they're restless, right? They're always moving, and this is the Brownian motion phenomena that is exploited in many fields. For example, there are commercial instruments available where particle size measurement is basically done based on this Brownian motion, for example, okay? This is actually, if you take these movies, right, and if I basically, sorry about that, right, if I take one such particle and track what they're doing, this is basically a trajectory of the particles. Okay, so maybe the particle was here, the next time instant, after a few seconds, maybe 30 seconds in this particular case, right? After every dot is basically 30 seconds apart. Okay, it's there, another 30 seconds is there, right? It's basically a chaotic path, right? This is actually the path that was actually captured by Perrin, which gave him the Nobel Prize, right? And what you can do with these paths, maybe some details. I think in the interest of time, what I do is, that's a picture of Perrin, right? And these are the beads that he used for his experiments. Okay, these are actually latex beads. Okay, these are beads taken from latex plant. Okay, plant has white sap, as I said, right? And he painfully separated. Okay, if you remember the picture that I showed, the particles were of very different sizes. But what he did is he came up with a way of getting similarly sized particles. Okay, and he put on a a glass slide like this, and he observed it under a microscope, and he captured these paths. And there's a nice theory that was developed by Einstein, okay, which related the mean square displacement of these particles to diffusivity. And if you know diffusivity, you can get particle size. And then there is some simple math by which you can actually calculate what is called Avogadro number, right? All of you know what is Avogadro number, right? 6.023 into 10 power 23, right? And one of the calculation of Avogadro number is based on these experiments. Okay, what you can do is, you can, because I know their paths, I can get the left hand side. That's your time. And this d is a dimension. Okay, it depends on whether you're doing experiments in 1 dimension or 2 dimension or 3 dimension, right? And then I know, if I know what is the size, right? So if I can get diffusivity from this experiment. If I know what is the size, if I know what is the viscosity, what's the temperature, I can get Kb. Kb is Boltzmann constant, and that is related to Avogadro number and the universal gas constant. Okay, so therefore what Perrin did was actually calculate, this Avogadro number. You see the Avogadro number calculation is about 80 10 power 22, 69 10 power 22. You see, I mean, it's very close. It may not be the exact 6.023, but this is an independent measurement that he did. He came up with a nice way of measuring Avogadro number that kind of proved the existence of atoms. Okay, until then, people kind of knew that atoms do exist, but then one of the ways of confirming the existence of atoms actually comes from the experiments of a colloidal scientist who was able to get an Avogadro number which is very close to what is accepted. Okay, yeah, maybe I'll just stop here, right? And if you have any questions, I'll be happy to take them. Yeah, thanks. Or any feedback, if you guys think that things were not clear, I can go back and explain a few things again. Okay, or if you have any questions, you could write back to me as well, right? I just mentioned my email ID. It's basa, BASA at iitm.ac.in. You can drop me an email, okay, or you can call me. I've shared my number as well in case if you have any doubts. I would be very happy to, as well. I'm also giving a talk day after tomorrow. Okay, I have a 1 hour lecture on some nice phenomena that you see every day. Okay, I just have lot of nice movies, and then I'm going to explain some interesting stuff. So I'll see all of you day after tomorrow. Yeah, and hopefully some of you will write to me and then keep in touch. Yeah, thank you. Thanks.
Thank you, sir, for that very interactive lecture. I'm sure it has piqued our interest into colloids, and we'll definitely research more about this. And thank you very much, sir.